This presentation will explore groundbreaking research in the field of prime editing for the sickle cell gene mutation. Our focus will be on the article titled, In Vivo HSC Prime Editing Rescues Sickle Cell Disease in a Mouse Model. This study represents a significant advancement in the gene editing technology, with the potential to transform the lives of those affected by this debilitating condition. As a brief overview, we will be discussing the research background, a description of the gene editing technology, research outcomes, and finally the ethical concerns.
Sickle cell disease is an inherited condition that profoundly alters the structure and function of red blood cells within the body. This genetic disorder is attributed to a single point mutation that prompts the hemoglobin molecules within the cells to bond together, resulting in the characteristic sickle shape, altered from their usual rounded form. Unfortunately, this deviation in shape increases the likelihood of blockages in the body's blood vessels, impairing the cell's ability to effectively distribute oxygen to vital organs and tissues. The consequences of sickle cell disease are far-reaching, with a spectrum of symptoms and complications. Patients frequently experience excruciating pain when these misshapen cells become lodged in blood vessels, obstructing the normal blood flow. Additionally, anemia may occur due to the reduced capacity of the sickle cells to carry oxygen throughout the body. Blood clots are another concerning issue, as they can lead to life-threatening conditions such as stroke and vision impairment by obstructing crucial blood vessels. To address this complex ailment, current treatment approaches involve the extraction of an individual's cells. These cells are further subjected to advanced techniques such as CRISPR technology to reactivate the fetal globin genes, or the reintroduction of lentivirals as vectors to modify the DNA. However, the primary objective of ongoing research is to rectify the root cause of sickle cell disease more simply and cost-effectively than the existing methods. The aim of our chosen study is to develop a gene therapy approach for sickle cell disease by directly repairing the genetic mutation responsible for the disease in the hematopoietic stem cells in vivo. An additional objective is to develop a technology that can substantially benefit individuals in developing countries, as they constitute the primary demographic affected by sickle cell disease. By limiting the need for cell extraction and lengthy incubation periods and transplantation procedures, this technology aims to provide accessible and efficient treatment to those in need. The hypothesis of this article is that episomal HD-ADPE5 max vector genomes would be efficiently lost in rapidly dividing cells. This is to be further explained within our description of gene editing technology. However, in essence, it means that the helper-dependent adrenoviral vectors can directly repair and correct sickle cell mutation in hematopoietic stem cells. In this study, prime editing is employed as the chosen gene therapy method. This technique involves a prime editor which will bind to both strands of DNA, cut it open precisely at the target site, and replace the target sequence or target base with the desired sequence. However, why is prime editing necessary? Sickle cell disease is caused by a substitution in the amino acid sequence at the 6th position of the beta-globin chain. Instead of glutamic acid, there is a substitution with valine resulting from a single base mutation. This missense mutation in the amino acid chain has resulted in the polymerization of the beta-globin. This results in the presence of the beta-S-globin gene instead of the beta-globin. This consequently leads to the beta-S-globin combining with the alpha-globin chain and producing sickle hemoglobin rather than normal adult hemoglobin. Due to this base substitution, this study employs a novel vectorized prime editing system, HD-ADPE5 max, a helper-dependent adrenoviral prime editor, to precisely target the desired beta-globin mutation. The adrenoviral vector serves as a carrier for delivering genetic material into target cells. Once integrated into these cells, the prime editor binds to the DNA strand and searches for the target sequence, enabling precise alterations. Prime editing is particularly advantageous for making minor changes to DNA sequences without causing full double-strand breaks, as seen with other editing tools like CRISPR. As these edited cells undergo division, they propagate the desired genetic change to future cell generations. In this case study, HD-ADPE5 max is tasked with performing thymine to adenine base substitution while introducing a silent base mutation from guanine to adenine. This modification changes the codon sequence to read as GAA instead of GTG, aligning it with the desired sequence for normal beta-globin. Furthermore, the choice of HDADPE5 Max over similar prime editors was based on its favourable carrying capacity for the genes to be transferred into target cells. To summarise, the innovative approach of employing prime editing specifically through the vectorized HDADPE5 Max system holds great promise for precisely correcting the genetic mutation underlying sickle cell disease, offering foundations to build upon for effective gene therapy interventions in the future.
The findings from the in vivo prime editing study highlight the remarkable therapeutic efficacy achievable through this approach. The study demonstrates a reduction of sickle cell hemoglobin, HbS, and a significant increase in the normal hemoglobin, HbA, within erythrocytes. This replacement of approximately 43% of HbS with HbA is a pivotal observation, underscoring the ability of prime editing to directly repair the pathogenic codon in hematopoietic stem cells using viral vectors. Furthermore, the study's results demonstrate that some mice experience near-complete correction of the sickle cell mutation, a profound improvement that showcases the potential of in vivo prime editing to not only modify the genetic defect at a molecular level, but also translate it into tangible clinical benefits. In order to assess the long-term effects of the genetic edits, modified bone marrow cells from primary mice recipients were further transferred into secondary mice recipients. A major finding from this process was that the proportion of genetically edited cells remained stable in the secondary mice. This suggests that the genetic edits occurred in long-term repopulating cells, known as hematopoietic stem cells, that hold the ability to self-renew throughout an organism's lifetime. This is a crucial observation, as it suggests that the edits were made in a way that is capable of long-term therapeutic effects. While these findings are significant, it's important to note areas for improvement to unlock future potential. Enhancing prime editing efficacy beyond the achieved 43% correction rate could yield greater phenotypic benefits. Additionally, while promising in mice, translating these results to human clinical trials involves complex challenges, including ethical considerations which will be addressed later in this presentation. Despite these hurdles, the improvement in phenotype testifies the clinical relevance of this approach and holds promise to bringing relief to sickle cell disease patients. Scientists propose that this research may have a substantial global impact, particularly in developing countries where access to advanced medical interventions is limited. A key impact lies within the potential for widespread application. Sickle cell disease remains highly prevalent in numerous resource-poor countries, spanning continents from South America, Africa, and Asia. This technique's minimal reliance on specialized equipment and laboratory infrastructure makes it more feasible to implement in regions with limited resources. The simplified and portable intravenous approach could offer a visionary alternative to conventional treatments that may be less accessible due to economic and healthcare limitations within developing countries. The use of HDAD vectors as delivery vehicles through IV injection adds to the approach's simplicity. Furthermore, this prime editing approach results in treatments that may be administered within the community itself, potentially reducing the need for patients to travel long distances to specialized medical centers. Compared to complex and invasive approaches towards sickle cell anemia, such as traditional bone marrow transplantation, the in vivo prime editing method offers a reduced burden on patients. Whilst bone marrow transplants traditionally require high-dose chemotherapy or radiation therapy, this prime editing viral vector approach offers a less invasive and potentially safer solution. There are 3 key ethical concerns surrounding this research that prompt a critical examination of potential issues and resolutions of this transformative technology. Firstly, the utilization of mouse models played a pivotal role in the methodology of this research through the intravenous injection into mobilized mice. While animal models are commonly employed to study diseases and potential treatments before human trials, it inherently raises ethical concerns related to animal welfare, consent, and autonomy. Addressing these concerns involves several crucial steps. Researchers should undergo comprehensive ethics reviews conducted by institutional animal care committees before embarking on any research involving animals. This process ensures that the research adheres to the strict ethical guidelines and minimizes harm to animals. Furthermore, researchers should actively seek alternatives to animal models whenever feasible. The development and utilization of advanced in vitro and computational models can reduce reliance on animal testing. Secondly, the translation of findings from animal models to humans is a complex ethical issue that arises due to inherent differences between species. While animal models have greatly assisted in advancing medical research, there are several factors that raise ethical uncertainty. Mice and humans are distinct species with varying genetic, physiological, and metabolic characteristics. These differences can influence how diseases manifest, how treatments are metabolized, and how the body responds to interventions. Furthermore, the translational gap between animal research and human application raises ethical concerns related to patient safety. Testing therapies in humans without a clear understanding of their safety and efficacy based solely on animal data can expose human subjects to risks. In order to address these concerns, researchers should engage in translational research, which focuses on bridging the gap between animal studies and human applications. This involves conducting intermediate studies such as human tissue or cell culture experiments to validate findings before moving to human clinical trials. Furthermore, ethical reviews of human clinical trials should involve a rigorous risk-to-benefit assessment, considering the limitations of animal models. These assessments should prioritize patient safety and the potential for therapeutic benefit. And thirdly, informed consent. Participants must be informed about the potential risks associated with gene editing, such as off-target effects, unintended genetic changes, or long-term health consequences. Simultaneously, they need a full understanding of the potential benefits, including the possibility of a cure for the sickle cell disease. As informed consent upholds the ethical principle of autonomy, participants must make decisions about participation free from coercion or undue influence. In order to address ethical concerns surrounding informed consent, comprehensive education is paramount. Researchers must provide clear explanations of the gene editing procedure, the nature of the treated disease, potential risks, and expected benefits. Informed consent documents should be clear, concise, and written in plain language. Furthermore, these documents should be reviewed by ethics committees and include all relevant information. In conclusion, by rigorously adhering to ethical guidelines, we can navigate the complexities of gene editing technology and move forward with greater confidence in the quest to alleviate the burden of sickle cell disease.